Okay, this is Chapter 3 lecture notes, starting with slide number 25, set A, so 25A. And we are covering membrane proteins. We talked about receptor proteins and transport proteins in the last set of slides. And so now we're going to look at proteins within the plasma membrane that function as enzymes. So we just call them enzymatic proteins. And an enzyme is an enzyme no matter where it is. So these just function to catalyze chemical reactions. And you can see here there's a chemical, this is a pair of enzymes. This is a pair of enzymes. So there's a green molecule that once it binds to the active site on the enzyme is converted to this red triangle molecule, and then the red triangle molecule this is like a cascade event, but is converted to the blue square, whatever that molecule is. So enzymes catalyze chemical reactions, and there are enzymes that reside in the plasma membrane. And then there are structural proteins, and you can look at this animation for structural proteins like collagen in slide number 26. And then we're going to move on to recognition proteins. Any of these proteins that have a carbohydrate chain, we call those glycoproteins. And they can serve as tags that are recognized by other cells, especially if the other cell is a cell of our immune system. So our cells of our immune system will recognize this as belonging to a cell that is a part of our body. We call it a self cell, S-E-L-F, self. So it will recognize it and not attack it. Okay. There's an animation on enzymes, slide number 28. And then we have attachment to the cytoskeleton and extracellular matrix. Elements of the cytoskeleton, which are structural proteins, help connect the cell to the extracellular matrix, which is going to be a substance that helps hold the cells and the tissues together. And then we have cell junction proteins, and these help join cells to other cells. These cell junction proteins contain CAMs, or cell adhesion molecules, and what they can do is they can allow the cells to adhere or connect to one another to form tissues. The glycocalyx consists of sugars or carbohydrates, which are chains of glucose molecules that are either attached to the lipids of the plasma membrane, and we call those glycolipids, or they're attached to the membrane proteins, and we call those glycoproteins. And like I was saying earlier, they can help in cell recognition. They can help the cells of the immune system to recognize that cell as self or non-self. If it's non-self, our immune system will attack it. And if it's a self-recognition protein, then our immune system will leave it alone, usually, except with autoimmune disorders. But that's another topic. So this is a clinical note on homeostatic imbalance. It says the glycocalyx of some cancer cells can change so rapidly the immune system cannot recognize the cell as being damaged. And so therefore the mutated cell is not destroyed by the immune system, and it's able to replicate and form into a tumor. Now, there are 3 types of cell junctions. There are tight junctions, desmosomes, and gap junctions. And these all connect cells to one another and help to form tissues. Tight junctions form a tight seal, an impermeable junction that encircles the entire cell and prevents fluid and other molecules from moving in between cells. So where might these be useful in the body? Well, one place is cells that line the urinary bladder. We don't want that stuff getting out into the rest of our body fluids, or the rest of our body tissues. So cells that line the urinary bladder are connected with tight junctions so that the urine cannot escape. And here is a picture of tight junctions. They kind of interlock and connect all along. Any cells that contact other cells, they just connect all along the areas where they touch each other. They're called tight junctions, and they form a continuous seal. And then we have the opposite of tight junctions. It's a junction, but it doesn't form a continuous seal. It's more of a very strong connection in one spot. A lot of times we use the analogy to a spot weld in welding. So it's a button-like connection, but it is very, very strong. And a lot of times the proteins in these desmosomes are called cadherins. And they interlock like the teeth of a zipper. Let's see. Desmosomes allow give between cells, reducing the possibility of tearing under tension. So where might these be useful in the body? One area is the cells that form the skin, especially as when the skin is shed every few weeks. The top layer of skin is shed and a new layer replaces it. Desmosomes connect the skin cells so that when we shed our skin, we don't shed just one cell at a time. We shed our skin in sheets. And it may even be easier to think about like when we have scaly skin, such as dandruff, that, we see it as flakes, which if we looked under a microscope would be many, many cells that are connected by desmosomes. Another thing is if we have a sunburn, sometimes people who sunburn can peel off some of their skin in sheets, and that's because the skin cells are connected by desmosomes. An area where this might be useful in the body would be the cells that line the heart, because what's happening in the heart is that it is pumping under great, great force and pressure. And so those cells need to be connected very strongly, very tightly to one another so that they won't explode or burst, So this is showing you a desmosome. Instead of being a seal that spans the entire area where two cells connect, that connects them everywhere they touch, it's just going to be like a spot weld. It's going to connect the two cells in one spot, but it's a very strong connection between those cells. Okay, so that's a desmosome. Then we have gap junctions, which are transmembrane proteins called connexons, and they form tunnels. They connect together and form a tunnel, and that allows ions to pass from cell to cell. This is used in cardiac and smooth muscle cells. The ions pass through from cell to cell so that the cardiac muscle and the heart can contract all at one time. So gap junctions are used for coordinated movement or coordinated contractions to make sure that all the cells are doing the same thing at the same time. And you can see how the connexons are connected to each other so that they form a tunnel through from one cell to the next. Okay? Let's see, we have movement across the plasma membrane, and this movement is either passive, that doesn't require energy, or it is active and requires the energy of ATP. All right, some types of passive transport are simple diffusion, and that means that the chemicals, the molecules or ions will simply diffuse through the phospholipid bilayer down their concentration gradient, which means from high to low concentration. Facilitated diffusion is the same thing, except instead of moving through the phospholipid bilayer, the substances will move through a transport protein. But no energy is used, and then osmosis is the diffusion of water. So we're going to skip some of this. So some factors that are going to influence how fast a substance is transported across the membrane would be the concentration difference from one side of the membrane to the other. The greater the difference in concentration of that substance on the outside versus the inside of the cell, the faster it's going to diffuse across the cell. The size of the molecules, the smaller, the faster that they diffuse. And then higher temperature increases diffusion. All right, molecules that passively diffuse by simple diffusion through the plasma membrane are lipid soluble, because the plasma membrane is a phospholipid bilayer, and so they have to be soluble in that phospholipid bilayer to pass through it. And nonpolar substances like the gases, oxygen and carbon dioxide are very small and nonpolar, and they can just diffuse through the phospholipid bilayer. This is called simple diffusion. And then facilitated diffusion is when larger molecules or polar molecules, or even ions that have a charge, will actually move through carrier proteins, and this is called facilitated diffusion. They're still moving from high to low concentration, but they are moving through a carrier protein. And then osmosis is the movement of water through the plasma membrane. So let's talk about in homeostatic imbalance. If the plasma membrane is severely damaged, substances diffuse freely into and out of the cell, compromising concentration gradients. Example, burn patients lose precious fluids, proteins, and ions that weep from damaged cells. So that is one reason we need the plasma membrane to be intact and not damaged. All right. So the substances that move across the plasma membrane through the phospholipid bilayer are nonpolar lipid-soluble substances. That means they're hydrophobic. Examples would be oxygen, carbon dioxide, steroid hormones, and fatty acids. These are substances that just simply diffuse across the phospholipid bilayer. And here's an animation of diffusion on slide 50. And this is showing you an image, an illustration of lipid-soluble molecules whose concentration is high. I'm just going to put an H for high. Their concentration is high outside the cell, and then an L for low, low inside the cell. So they're just going to simply diffuse through the phospholipid bilayer from their high concentration to their low concentration. Facilitated diffusion occurs with certain molecules like glucose, amino acids, and charged atoms or ions that have a positive or negative charge. They are hydrophobic. Sorry. It should say hydrophilic. Okay, this is a mistake. I just happens sometimes. It should say hydrophilic, P-H-I. See if I can do this without making a big mess. Hydrophilic molecules. Glucose, amino acids, and ions are hydrophilic. And well, some amino acids can vary, but glucose and ions definitely are hydrophilic, and they can either travel across the plasma membrane by carrier-mediated facilitated diffusion. That means that they actually chemically bind to a protein carrier, or they move through a channel protein. But it's facilitated diffusion because a protein, a membrane protein, is being used to transport them across. They don't just travel through the phospholipid bilayer. In carrier-mediated facilitated transport, you have a carrier. and it will transport specific polar molecules like sugars and amino acids, and they bind with the molecule, and after binding with it, they change shape, and then they move the molecule across the plasma membrane.